sponsors today are 3M, and it's really so great to have them on board. We're doing a series of Talkshop Fridays with them, and so this is number 2. So it's so exciting to have them back again. The first presentation was really, really interesting, very, very helpful in terms of helping you to make decisions about new technology. And I'm sure today is going to be no different. So thank you so much to 3M for their generous support of today's Talkshop Friday. So the topic for today is using artificial intelligence to improve your lab productivity. Ultimately, I think we would like to get results out as quickly as possible, and we're stretched with resources. Everyone's looking at costs. So how can we do this in the most economical way, and also make sure that we've got the best accuracy, and not just using traditional methods, but actually using technology to assist. And so it's really interesting that today we're going to be talking about the 3M Petrifilm advanced plate reader, and how this actually works. And Louise is going to be taking us step by step through this process to understand exactly how this piece of equipment works. So this is almost like having her in your lab doing a trial for you. So make sure you ask all the questions you would ask if she was actually able to be with you right now. So first of all, who is Dr. Louise Mare? It's a real honor for me to be chatting to her today because I've known her for a really long time. She has a PhD. Hi, Louise, microbiology, technology. She's worked in the food industry for a really long time. She's been working for 3M for the last 11 years, and she's provided a variety of roles when it comes to new products, customer training, marketing, management of global key accounts. Before that, she was really in research at the Agricultural Research Council, and she has a lot of experience from that point of view. So the point being is that she's worked in a lab and she knows the struggles and the pains that you have in a laboratory environment. And so hence she's the perfect person to work as the technical sales and technical manager for 3M, the food safety division. So Louise, I know that you would much rather be fly fishing today. I know you're with us, but after this, I know you would like to go fly fishing, even though it's cold. Maybe today is the right time to watch a movie or read a book. But before you do that, we would love to hear all about this particular product. And so I'm going to hand over to you now, and we're looking forward to your presentation. Thank you so much for being with us today. We really do appreciate it. All yours.
Thank you so much, Linda. I hope that you're going to enjoy today's presentation. And yeah, please, like Linda said, feel free to pop in as many questions as you like, and we will see how much we can answer. Otherwise, we will definitely get back to you. So the exciting news is that 3M has launched a new Petrifilm plate reader, which has got features that is extremely unique. And I'm going to take you through a little bit of a journey about this plate reader. First of all, just as a little bit of a background for those of you who might not be all familiar with our 3M Petrifilm way of testing. This is just a slide to show you Petrifilm is basically the same as an agar plate, right? So when you do a sample, you've got your piece of cheese, you want to know how many bacteria are in the cheese, you basically dilute it in a 1:10 dilution, you plate 1 milliliter of the sample onto our Petrifilm, and you incubate. And then what you will see are these little red dots on the Petrifilm. Those will be the colonies. Now, the colonies needs to be counted. So there's 2 ways to do it. You can either do it manually. That means you use your eye, your human eye, to count it, or you can use our advanced plate reader for this purpose. So this is just a slide to show you exactly a picture of when you inoculate the plate, you then incubate the plate, and then all about the interpretation. And that's what we are talking about today, right? So 3M basically went to the customers, that is you, the guys in the lab, and said, what do you want in a plate reader when we developed the new one? So increased 3M plate functionality. That means our previous reader, for those of you who might be aware, could only read 6 of our Petrifilm plates. The new one can actually read 10 plates, and that is really a big improvement in functionality. And as 3M develops the software and the technology, of course, we aim to actually cover the full range of 14 plates. So there's only 4 plates that's currently not yet readable on the new Petrifilm plate reader. We want consistent and accurate enumeration. Of course, that is very important. You want to know when you get a count from the plate reader that it is really accurate. Fast time to result. This new plate reader is extremely fast in calculating the results. Then result trending and reporting capabilities, and this is where the artificial intelligence basically comes in that I will talk to you about. Easy to use, that's always important in the lab. You don't want to read a very thick user manual to be able to know, how to use the machine, and then designed for easy cleaning. So which plates actually can be read in the new Petrifilm plate reader? So these are all the plates that currently you are able to read. It's the total count, coliform, Enterobacteriaceae, E. coli, coliform, select E. coli, the staph, and then also the staph press with the confirmation disc added. Then a rapid aerobic count, rapid coliform count, rapid E. coli coliform, and rapid yeast and mold. And that's the very good news, is that even the rapid counts can now be read on the plate reader. So the 4 plates that can't be read yet is the normal yeast and mold petrifilm, the lactic acid bacteria plate, the high sensitivity coliform, and environmental listeria. So those are the 4 that remains that needs to be added, and eventually I'm sure that they will be added. This is really a machine that's developed for consistency and accuracy. It actually gives you a high resolution image capture. In other words, when you put your Petrifilm in the machine, it's going to take a photo of that specific Petrifilm, and this will be a high resolution image. Onboard fixed artificial intelligence networks, and I will talk about them a little bit later. Trained with a wide assortment of global foods. That means in the machine, when the machine was developed, so many samples of any food that you can really think of was actually used to develop the sort of databases and reference material that the machine used when they actually then pick up that you are reading a total plate count from milk, for instance. It actually then, that is a little bit of the artificial intelligence where it actually goes back to all the memory that it's got of any milk samples, and it sets the machine. There's a little adjustment that happens to adjust for that sample. And that is the artificial intelligence as well. It adjusts a little bit to be able to read the dairy samples very efficiently, just as an example. It's driven by powerful software and counting equivalent to a trained technician. But of course, you can never take away the human eye. Remember that this is just a machine, so therefore if you see some differences on the Petrifilm, that the machine is maybe not counting 1 or 2 colonies, there is a way that you can even edit and add those colonies. So we want to improve lab efficiency. Results is in 6 seconds or less. Now that is really, really a good improvement from the last machine, and it is very fast. You can also stack about 24 Petrifilms in the loader, and they will just push, the machine will just push it through very quickly. You can enumerate up to 900 plates per hour. So that reduces your interpretation time by up to 94%. And I know that so many people spend hours counting the colony. So if you can reduce that time, it definitely frees you up to do any other work that you might never have been able to get to. This machine also recognizes 10 different types of barcodes. This is a great added advantage because the companies that use barcodes, a lot of information are embedded in the barcodes. So immediately if the machine scans the barcode, it picks up the sample, it picks up maybe the date, it picks up what any background information about the sample itself. And you can create work lists to optimize the workflow, which I will discuss also a little bit later. So as we mentioned, the software is really extremely advanced. The first step is enumerate. That means it takes the picture, counts the colonies, basically. The second one is it reports your results in various ways. And there's such a lot of extras in this report function of the results that you can use, and this leads to the trends and reports in the end. You can do bar charts, you can do pie charts. There's extremely good amount of charts and graphs that you can actually use to do your trend analysis and reports. So the nice thing is also when the machine is started up, it basically does a self-check. And that's very important because you need to know that whenever you're using the machine, that it is going to be accurate. Okay, so there's a built-in self-check that every time when the machine gets started up and used, it first will do the self-check to know that everything is still in order. The nice thing is it looks really good, this machine. On top of the machine, there's a color-coded light bar. So this color-coded light bar, the different colors actually also mean something. So upon powering the device, you'll have a white light for about 25 seconds. After the 25 seconds, it will turn blue, and it will then remain blue until you have launched your software that goes with the machine. When it is green, it means it's active. It means you are busy reading your plates and your software is open. If there's an error, like a plate jam or something, or the PC application is not running, of course, it will be a red light. So this is really nice feature as well, one of the nice features of the machine. So basically, you insert the plate with the inoculation zone facing up and the hinged portion of the plate facing the entry slot, okay? That means you put the Petrifilm in, the camera is on the top part. It's going to take a photo from there onto the plate. You must just make sure that there's not excess moisture on the plate. And I'm not sure if many of you know this, but you can actually freeze Petrifilm and look at them again later. But if you want to read them again in the Petrifilm plate reader, you must just allow for them to thaw before feeding them in again. Okay, so on the enumerate now screen, there's actually 4 ways that you can feed the Petrifilm plates through the Petrifilm plate reader. So the first one is the plate top drop-down menu. You again will have the function like in the previous plate reader where you select which plate type did I use. So was it total count? Was it Enterobacteriaceae? So you basically select this plate type, okay? The users also have the option to select the lock icon if they are running many plates. That means as the same plate type. So every time then when you feed the plate in and you're doing, let's say, 50 total counts, you just use that little lock function and you don't need to select every time the type of plate. Copy to Excel is really nice, okay? This is used in conjunction with your plate type that you pick, or even the barcodes. That means once the plate goes in and fed through and the result becomes available, it automatically generates an Excel spreadsheet. So this is also a bit of the artificial intelligence where the machine immediately knows then if you've picked this feature, okay, sample is being read, I'm going to generate the Excel spreadsheet, okay, with all the information in there. Then barcodes, as I mentioned, there's up to 10 different types of barcodes that can be used at the moment on the machine. And this is a feature that is really growing because people don't like to write the sample ID and everything, as it also takes a lot of time. The extremely nice thing is Worklists. So what is Worklists? Worklists is if you have a certain amount of samples that you want to do with the machine, but it's regular samples. You can create a work list and feed it into the machine, that the machine already knows if you go to a certain work list that you've given a certain name, it will know exactly that it is rapid yeast and mold that you're counting, it is these specific samples, you're using this dilution, and the temperature is this, that is the temperature that the machine is running at, and basically it generates all that information automatically into the machine, and the machine will know when you open the work list exactly what you're going to read. On the results screen, there's many features that you can use. You can do various searches. Let's say you want to see all your results on a certain period. As you can see, there's a date range here. Let's say all your ground beef samples filtered through a certain date. You can do that. Also, you can basically use other types of filters. For instance, apart from the date range, you can use what dilutions, you can use the sample ID itself, or then, of course, the product name. Calculating the results. So the software can calculate the CF used based on a single plate and a selected dilution, or multiple plates and multiple dilution. So it's a single plate at one dilution, or multiple plates at one dilution, or multiple plates at multiple dilution. So it's very easy to use whichever plates, whichever dilutions. You just basically select from a drop-down menu. Exporting the results. As I said previously, there is the Excel spreadsheet that you can immediately generate. It can also be exported to CSV format, TSV, or a custom delimited format. And remember that the machine is LIMS compatible as well. Now, the important thing is we need to use our data. We don't just want to do a count. We want to work with what we are getting as a result. So it allows you to release the product. So we know, first we collect the sample, then we record the results, then we transfer the results, basically from the plate to the machine where the results are reported. But why do we want to stop there? The important thing is the second block. We want to analyze those results. We want to visualize. That means we want to maybe go back to that Petrifilm picture and see what actually happened there, how does the colonies look, and then we want to take action. That is a very important thing that we need to do. So yet again, just to recap, the technician analyzes the sample, right? The data is passed real time to the software. The manager identifies a spike in the counts or something, and corrective action can be put in place immediately. Report generation. As I said, there's so many ways that you can generate the report. You can basically click from a drop-down menu. You can have all of these functions and even more in your report. So you can have more or you can have less, depending on basically what you need. You can also set up real-time charts. So you basically establish how far back you want to pull the results. So it is a moving window. In this case, it will also be, let's say, the last 6 months from the day you are looking at the chart, like in this example. You select your filter material. That is, what do you want to look at? This is the part where you filter. And then you basically you go to add real-time chart. You can name the chart, and when the software then eventually draws up the chart for you, it allows you to determine if your new results are actually in line with the historical data, or if there are some irregularities that may require some sort of a corrective action. So this is also part of the artificial intelligence of this machine, as to knowing, and being able to actually get results from 6 months together within easy clicks. Recurring reports. Let's say there's a report that you need to do monthly or weekly or yearly. You can set up a recurring report that it generates automatically. So this is an extremely nice feature, especially for audit purposes or for when you have meetings with quality control or in the lab, or even for managers, etc. And so you don't need to take time each week to pull a report. It basically does it by itself. Okay. And yeah, that is our new Petrifilm plate reader with a lot of excellent features. And it is actually a machine that is really well liked on a global level. And we are really having a huge successes with our plate reader. And I'm really excited that it's going to be launched, or it is launched in South Africa. So it is available currently. There is a little bit of a delay on the lead time, and because there's such a big demand, but we are always here to assist you. And yes, now for questions, if you have any questions.
Thank you, Louise. That's great. I think in terms of the, what's the possibilities out there and what have you, as to how this can assist, the mind boggles. Just to think of how simple we are now able to make this process. I think even I could do it. Which definitely says something, because when I was in a company where there was a micro lab, I was banned from the micro lab because I was not considered a competent technician. So Louise, I've got lots of questions, and I'm sure everyone else does as well. So would you mind putting your camera on so we can chat to you and ask you these questions? That would be so cool. So at least we can have a bit of a face-to-face conversation.
No problem. There we go.
Yes. Okay, so the first and probably the question that's burning on everyone's mind is, how much does it cost?
Yeah, I know that is probably the question that a lot of people ask. So we've got different pricing structures for different customers. So it's difficult to say one price, because we've got, of course, it depends on volumes that you do in your lab as well. And we can look, we really want to get the machine into as many labs as possible. So there is even options where we can do a rent to own option, where we can basically put the machine in, and then you basically pay it off over a period, etc. But the cost itself, I always am hesitant to talk about the exact cost, but so I think rather a ballpark figure, and unfortunately it's in euros because the exchange rate to South Africa, but the fixed rate is 6,600 euros.
Okie dokes. All right. So that means I just wanted to know whether I would need to sell 1 kidney or 2 kidneys. You know, sort of a ballpark figure.
Yeah. So that's it.
And I'm sure you had done the sort of return on investment calculations. So you know, when dealing with a company, you can actually show them the payback period and all of those things.
Yes, definitely. Yes, so that's why I say the fixed global cost is the 6,600 euro, but then we work from there. You know, we can always do a deal.
Super, right, everyone, you heard that. So, and we've got her on camera and we have her on recording. So you can also see that, but obviously please, you know, engage. And of course, 3M will also be contacting you as part of this webinar to pass on some more information. So I really like the lazy person features, and I say that because you know, I think sometimes the detail on samples and those kinds of things, that they're really critically important. And if we're not transcribing that information correctly from one piece of paper to the next piece of paper, or even the LIMS system, we can actually lose traceability of the sample, and particularly if it's a corrective action involved in a corrective action process. I really like that. What I was intrigued by, Louise, was, if I understood you correctly, the system already has some data in it with regards to certain products, food products. Am I correct?
Yes, absolutely. And that is the artificial intelligence part. So it's really incredible that they could manage to do this. But because we had the previous plate reader, which analyzed a whole lot of samples, they actually realized that, let's say it's a dairy sample you're looking at, the machine needs to focus its machine eye in a certain way for a specific sample. So immediately when you put in that it's a beef or a meat or a dairy or whatever sample, the machine adjusts itself according to data that it has built inside, to be able to really analyze those samples absolutely correctly for you. And that is such a unique feature because, yeah, it really is that information in the background that makes it so great.
Now that can be a pro, definitely, because does that allow me to sort of, I mean, is there a benchmarking feature, or I mean, am I able to compare my product with, you know, that data that's already in the system in terms of specifications and sort of generally acceptable counts?
No, not really, because it all happens in the background. So this is specifically the machine's eye adjustment that happens. So it's not really going to be able to allow you to compare your counts with historical data or something like this. But what it will do is it will adjust that machine's camera or focus to be sure that you get the most accurate count, if you understand my answer.
Yeah. No, I understand, because I think it's almost like when you have a situation, I understand within my little bit of micro in terms of making sure that you're accredited based on food matrices. So I think in this way you're compensating for for the food matrix.
Exactly.
In order to be more accurate in that regard. I really also thought the barcoding aspect is great, because like you said, there's so much information that we're already capturing on a barcode. Why would we want to repeat all of that information? You know, by just using the barcode. I thought that's a really neat feature. And then one of the other questions I just wanted to ask you was in relation to the corrective action process. So you said, you know, you showed the cycle where I read the plate, the petrifilm rather, we identify a spike, and then corrective action takes place. Does that have to then come out of the system? So that's got to go into another system for us to track the investigation and what have you. That's not built into the reader?
No, so basically, you can actually, when you generate when you use that feature where you generate reports at a certain time, let's say once a week on a Wednesday, automatically the machine generates a report for you. So that is part of that feature where you can then basically immediately see if it differs hugely from what happened last week, for instance. And you can actually click on a graph and just put dates in, let's say, so you can put a certain month. You can do it over a month, weekly, then you can generate that report and it will immediately do a trend analysis for you to see if there was a spike somewhere. So I don't know, does that answer your question?
Yeah, but so I think just from a, in terms of the next step, I'll have to take that report and I'm going to use that data then in my investigation, which will probably be on my corrective action system, which is outside of this. And then I would then possibly retest results. I would be able to then, I'm going to have to retest them. I'm going to have to read them again. So then I would be able to show, that the loop then on the
That's
reader.
right.
And possibly the sample identification. I can then, because I don't want to lose that sample again. So the way I identify that sample to link it back to the corrective action investigation for the trend. Okay, so we've got some questions coming through. I'm finished with mine. So now everyone, please, Christina is sending questions. So everyone, please, this is your turn. So you're welcome to. So what about updates? Is there? We're assuming this is connected to the cloud in some way, and so that when 3M has updated information, or it kind of talks to the reader. Is that how it works?
Absolutely, yes. So basically, if there's a software update, because obviously there's improvements that happen all the time, and let's say there's another type of Petrifilm that gets added to the range, you just go onto the you will get an email notification that there's a software update available. You just go onto the 3M website, you put in your serial number of your machine just to be sure that you do actually have a machine and you are using the software. And then you update the software for free. There's no charge for any updates of software. And yes, the software is, updated as they go along. Yeah.
But now talking about that, we have to all think about this Papier thing, okay? So now what about, what if we do unfortunately have some bad counts and, because now obviously if I'm talking to the cloud, it can go two ways. So 3M must be able to access my results. So how do we know that situation is protected?
Yeah, I don't think that there is actually, because it doesn't go into a cloud that 3M controls. Okay, so when you've got the software loaded on your system, 3M doesn't have any access to the results. You work with it on your PC and on your internal database. Okay. So we're not really going anywhere near your data or your results or anything as far as that is concerned. Like I say, we just do the software updates, but we don't have access to all the softwares running in the world and what their results are. No, we definitely don't go in there.
Okay, that's good. But that also means I've got to make sure we've got appropriate backups and those kinds of things. And I'm sure you advise when we set it up so that we don't, we don't think that it's going into a cloud, but actually it's going into a pink cloud that's just going poof, and it's gone. All right, so can I then determine the high and low limits? In other words, my specifications. Do I input my own specifications for the sample type so that it immediately flags or alarms if there is an out of spec result based on my specific specification for E. coli, for example?
So this is a feature that I think is coming in the future. It's not yet there. We've done it with our clean trace system, where you can basically put in your limit range that you want to measure at, and then if it goes over or under it, you will get notified. So it's not yet there, but it's definitely something that they are looking at.
Perfect, because I think that would be an extremely useful feature. Again,
Yes, definitely.
just making sure that we're not missing anything. You know, yes, you obviously don't want to exclude the management review of the results and the action, but I think it would be really great if it could highlight exceptions, you know, rather than just all the results. Okay, last question. If I'm understanding you correctly, Christina, so you said that the first thing the machine does is a self-check, so make sure its brain is still working. That's good.
Right.
But from a QC perspective, I think if I'm understanding you know, how do I make sure that it is really reading the right colony? You know, from that perspective, I know the Petrifilm is selective, but is there any additional quality control? Do I need to use a strain that I have to specifically, you know, inoculate with to double check that it's actually reading the salmonella colony correctly? Do you understand what I'm saying? Christina, I hope I got it right.
So with our previous Petrifilm plate reader, we had what I called a verification card that you basically insert into the machine before you start, and it checks whether everything is still in spec. So whatever, if you are reading a non-environmental, a rapid yeast and mold, for instance, you know that the results that you're getting will be accurate, etc. So with this one, with the self-check on startup, they've actually built in that feature that it actually goes and checks a lot of the points, as I showed in the slide. It's not only one thing that it checks, it's actually a lot of points that it checks and makes sure that everything is working properly, everything is functional, and there's no need for you to do like a pure culture. Let's say you're checking for E. coli count, then take a pure culture, put it on a Petrifilm, read it to make sure. There's no need for that, because like I say, the data that's running in the background of the machine already has so many pictures of E. coli colonies in there that it adjusts the machine to give you the best possible result by adjusting the way that the machine looks at the colony and recognizes the colony. As I said, remember, it's only a machine in the end. The human eye is always necessary. So let's say for some reason you see on the picture that there's a colony or one or two colonies that the machine didn't read. You can actually go and edit. So you can go and add those two colonies. Okay. Or let's say there's a little speck or something that the machine is now counting as a colony. It can be a food particle. Yes, the intelligence of the machine is made that it shouldn't read particles. It shouldn't read something else on the background, but it can happen. Then you can actually go and unclick that specific reading and edit it to say, no, this is not a colony, this is something else. So yeah, the human eye is always just, there for making sure.
And I assume that those kinds of editing rights would be restricted with a password or something, because
definitely. I don't want to. No, you don't. No, you don't. You don't really want. Only certain people will have that function to be able to edit anything. And yeah, it's also very controlled, because once somebody has edited something, the machine knows, the software, who's logged in and who edited it. So that even goes into your Excel spreadsheet where your results are reported. If anything was edited, it immediately shows there.
Okay, so that's good. Any auditors that are listening, I think that's important, because it's an audit trail to ensure the integrity of the results. So I think it's important that we understand, the safeguards because we don't want to be manipulating data that can ultimately impact on the safety of our
No. And what also is nice, remember, if a certain Petrifilm was edited quite a lot, many changes, you can always, when you have an audit and the auditor say, well, I want to see that Petrifilm, the picture is always available. So you can basically go back to the image that machine has taken, show it to the auditor and say, okay, this is the reason why I edited this. And
Absolutely.
so that's easy to just, get the picture from the software.
Yeah, I mean, that's just amazing, just in relation to all of those Petri dishes that then get destroyed, and there goes the evidence, and we never see them. Whereas here you have every single that you're counting. So like you say, you can go back to the raw data. That's absolutely amazing. I think I can actually now apply for a job in a lab. I'll actually be able to do it. I won't make so many mistakes. The thing
I'm
is,
sure you will.
Okay, so I think that the challenge with when we start to use artificial intelligence is that it requires such a high level of technician when the thing breaks. It's all very well for Linda to push the sample in and press the button, but it's when it breaks that we have to bring in the real brains. So Bilal wants to know, how frequently does it need to be serviced, and if there is troubleshooting necessary, then we have to bring in somebody, from in to assist, yes?
Yes, so currently it's like all lab equipment. We actually do specify that it needs to go in for a calibration at least once a year or for a checkup. And we are working together with our service agent CSX that is countrywide. They service all the medical devices in 3M's medical division, where food safety actually also resorts under the medical division. So they already service our LM1 ATP luminometers. So that's a nice thing, is that it's a local service, and we are now in the process of getting them set up, going through the training to be able to service all these machines locally. If parts needs to be replaced, they can do it, etc., etc. And what was the other part of the question?
No, I think just in terms of training, from a training for a technician, but also if it breaks, I'm assuming CSX will be the ones that we bring in. So they're not just for calibrations, but they're also then for maintenance if there are issues. Okay.
Absolutely, and the turnaround time is actually pretty fast. And as we now sell more machines, of course, we will have demo units or standby units available, because we understand that the customer can't really be without the machine for too long, or actually we need to give them something that they can continue working. And yeah, that's the way that we want to, do the service part.
Super. Thanks, Louise. Okay, well, that sounds great. I'm really, really impressed. I think this is a wonderful, machine, and I sincerely hope that your trend graph for 3M looks good, in the sense that it's going to be going up and up and up and up when it comes to sales, and that more and more of these units will be available. So thank you very much, Louise. Thank you for your time. We appreciate it. Thank you, 3M, for coming on board. For everyone who's listening, you can definitely expect to be hearing from 3M so that they can give you some more information about the product, so that you can evaluate it. And I'm sure you'll have more questions for Louise. So thanks so much for being with us. We appreciate you being with us today. Enjoy the rest of your Friday. Have a lovely weekend. Thank you, Louise. And everyone,
thank you.
Stay safe. Stay safe.
Yes, definitely. Thanks, everyone. And thanks, Linda. And I hope you all have a very good weekend, and please stay safe. Thank you.